Diffuse midline glioma is a rare aggressive brain tumour that occurs mostly in children. For 40 years, it had no targeted drug. The first, dordaviprone, arrived last year, tested on only 50 patients because 50 is the size of the whole field. A team working on a cancer this rare is not short of published research. It is short of the budget to test every path, so the next experiment gets chosen on judgment, and a wrong call costs a year at the bench. That decision has been a six-figure service sold only to the largest drug companies. I built Onkydra so a seed-stage founder chasing a rare cancer drug can make it too. You ask a question in plain English, and Gemini turns the sentence into a bounded plan before anything runs. It shows the indication it matched, the target it pulled out, and the 5 stages it will run. You approve the plan, and it starts. It fits a model to 60 real patients, then knocks out genes in a network of the tumour to see which interventions actually shift its biology. The answer ranks the tumour cell states under the OLIG2 knockout. The OPC-like program collapses hardest. The lineage OLIG2 governs, and it holds even when you strip out OLIG2's own expression. So it is the network responding and not the target counting itself. It ranks the patient subgroups, and it names the escape routes that would open next. Onkydra does not only work on the molecular data. Handed the tumour's own slide, and MedGemma, a medical vision model, running live on a GPU, reads the histology in seconds and describes what it sees. It reports the morphology, and it never offers a diagnosis. And every run ends with an Honesty Critic, which decides whether an answer is even fit to release or requires a human-in-the-loop intervention. This one cleared with a few points flagged for the user's judgment. An hour earlier, and the same critic held a different run and kept it until the user reviewed it. And that was one question. Behind it is a whole workbench. A state atlas of the tumour at single-cell resolution, a model match that finds which laboratory lines resemble the patient, a perturbation lab and a matrix to sweep targets at scale, an escape map for resistance, and an uncertainty lab for what the model cannot yet know. And all of it stays traceable, an evidence trail, a provenance graph, and a change history of what moved and why. None of it is a black box. And it doesn't end with a document, it ends with an experiment. The exact protocol, the prediction, and the result that would prove it wrong, locked into the record before you run it. When the bench result comes back, it's filed against that prediction, so the record becomes a living lab notebook of what was predicted and what actually happened. Do that across enough cycles, and you have what rare cancers almost never get: longitudinal data on the disease itself, the beginnings of a digital disease twin that sharpens every time a bench result is checked against it. Onkydra is the target decision that costs 6 figures, now in the reach of a rare cancer lab.